I think we'll get started, and hopefully the others who signed up will join us. Cindy, you'll monitor requests to get in, I hope. Welcome and thank you for participating in our first McGraw speaker series to be done via Zoom. We're happy to do it, and I believe you're in for a real treat tonight. Our speaker is Dr. Stanley Gehrt, who is the director of the McGraw Center for Wildlife Research, but he is also the professor at The Ohio State University. And Dr. Gehrt has been working for more than 20 years with McGraw, and he is best known, and I will say internationally known, for his work with urban coyotes in Cook County and in Chicago. He has spoken to us frequently in the past, updating us on the research that your membership and support of McGraw helps to foster. But tonight we're taking a little bit of a different tack, because not only is Dr. Stan one of the finest mammalian predator scientists in the world and world renowned for that, he is also at the cutting edge of how technology is changing the way we monitor and learn more about our wildlife. And as a result of that, McGraw is on that cutting edge. And that will be the topic of tonight's discussion. We will we'll have an opportunity to ask questions towards the end, but with that, I'd like to turn it over to Stan and say thank you for joining us, Stan.
Thanks, Carrie, for having me. Can you hear me okay?
Yep.
Okay. Yeah, with the number of people that are here, I don't know if it's, I guess it's still probably better to wait till later, but I would encourage someone, if I am covering something that you really have a burning question about, I'm perfectly fine with interrupting. I mean, go ahead and shoot right in there. I think you can do that. I think it's shared for everyone, maybe? I'm not sure. If you can, feel free to jump in. I also have to say, I've been staring at a computer monitor all day. I'll be so thrilled when we don't have to Zoom these meetings. So hopefully that'll happen fairly quickly, where we can finish this. So my eyesight's a little strange. I do have to say that Charlie Potter has never looked better, never sounded better than he has tonight. But it looks like his hairstyle has changed a little bit since the last time I saw him. So I will share my screen if it's okay with everyone. And as Carrie mentioned, I'll be focusing, rather than a particular project, I'll be focusing more on the technology that we use in different investigations. So let me do this. Does that work for everyone? Can you see my screen? Is it good? Okay.
Great, thank you.
So before I start, I just want to mention that a lot of the things I'm going to be presenting, I'm basically these days more of a paper pusher than much of anything else. I seek the funding for a lot of this, but the people that are down on the ground and actually charged with executing some of these crazy ideas are primarily Shane McKenzie, who's the McGraw Research Associate there on the ground there at McGraw. Shane has been working at McGraw for a number of years. We brought him in as a technician many years ago, and he stayed and has become an expert in his own right with some of the technology we've been using, especially GPS radio collars, which I'll be mentioning a little bit later. And then Dr. Charlotte Milling is the postdoctoral research scientist that is associated with both Ohio State and McGraw. So her job, she actually lives out in Montana, but she's an expert in quantitative skills and certain types of analysis. And so she's become pretty important on some of these projects, especially the one in Manitoba that I'll be talking about. And that's her trying to radio track some raccoons That's actually during the spring in Manitoba, but it doesn't look like it. So this is going to be kind of a brief outline. I had a hard time kind of narrowing this down to a short list. And some of these I'll go through more quickly than others, but I'll start off with one of our older studies, but it's actually one of the most impactful studies we've done. And it was clearly cutting edge technology, and it's proximity detectors. Then I'll talk about GPS tracking. So in that case, we're using radio collars that are monitored by satellites as opposed to us physically. And then stable isotopes is something that I really brought into terrestrial systems, and especially mammalian carnivores. And that's a technique designed to evaluate diet. And I'll show you what that's like and how we're continuing to use it in more and more different ways. So we're still kind of staying near the front to that technology. Drones we'll touch on as a type of, it's obviously you know what drones are, but it's being used in research by Delta Waterfowl, one of our collaborators. And this is something that's been funded by McGraw. And then I'll combine stable isotopes GPS in that Manitoba study. And then cameras, remote cameras in particular. And then, yes, that says vaginal implants. So we'll try to keep this a PG rated presentation, but we do use vaginal implants for white-tailed deer. I won't have time to get into genetic analysis, but there's new genetic techniques that are being developed all the time. And we're using genetics in some of our research, but I don't have time probably to get into that. But if you have questions, I can definitely address it. So proximity detectors. So this picture is actually an early study back in 03 and 04 of raccoons that we painted, and we turned them from nocturnal animals to daytime animals so that we could videotape them and identify them based on the color and where they were painted. And so the question would be, well, why were we doing this? We spent a couple of months on this and then a full year evaluating the collars that are on these guys. And these are true radio collars in the classic sense, but they had another feature that had never been done before, and that's the proximity detector. So what is that? Well, this is our attempt to measure contact rate between individuals. And that's a very difficult thing to measure for free-ranging wildlife, especially if you require an estimate of how often two individuals come into contact with each other at any time of the day or night during a 24-hour period. If you can't see them, that's almost impossible to do. And unfortunately, for nearly all wild mammals, it was previously an unknown factor, and yet many diseases, important wildlife diseases, and important diseases in some cases that spill over to people, rely on contacts, direct contacts for transmission. And so contact rate is actually a crucial parameter if you're going to try and model and predict the spread of a disease in a wildlife population. And in our case, we were looking specifically at trying to estimate the spread of rabies in raccoon populations, because raccoons are a major host of terrestrial rabies in the US. So these are just a few of the diseases. Some of these you'll recognize: chronic wasting disease, obviously, and then distemper and parvo are the viruses that you get your dogs vaccinated against. And so by pure happenstance, we were doing this work with raccoons. We were trying to model rabies in raccoons without actually knowing the contacts. And I happened to run into an engineer that actually works with wildlife telemetry equipment, and he had just patented a new technique to use UHF as a communication signal between transmitters. And yet he hadn't been able to use it or market it because he had no system to actually apply it to. And I just, by luck, happened to talk to him and said, well, we are working with raccoons and rabies, and this is the perfect system for that. It's actually the right size transmitter for those animals. And so we got right to work on it. And these were the prototypes that we had to evaluate because they'd never been placed on a raccoon before, or any animal for that matter. And so we needed to verify that they were reliable. Once we verified they were reliable, then we were able to put them on wild raccoons over in Cook County, one of the forest preserves. So this is kind of grainy image of, this is one of our spy cameras that we set up. And you'll see a flashing light under the neck of these individuals. These are those radio collars that have the proximity detector, and every time it flashes, it's detecting the presence of the other individual, and it could record the identity of that individual, the date and the time of that contact, and it would also record how long that contact occurred. And so this had never been done before. Here's after our initial research, we set up feeding stations to try and manipulate contact rate. We were testing certain hypotheses. And so here's a feeding station. Immediately within 24 hours, you had 9 raccoons using that. And you'll notice, again, the flashing light on these. So this is our study population after we got the original data. We monitored them for a year before we began manipulating them. What we didn't realize, because raccoons typically, if you're not feeding them, if you're not putting out feeding stations, they're not necessarily known to be that social. We didn't realize that they really are contacting each other a considerable amount. So this was, we put the collars on 40 animals and monitored them for 9 months. And this is just what we call a social network. And so this was the first attempt at trying to characterize the contacts within that population. And for example, each one of these nodes represents an individual raccoon. Some of them are circles, some are squares. That means some are females, some are males. I just highlighted 4 in the middle because the location of the individual within that network basically illustrates how well connected they are to the other individuals. So obviously the lines connecting them represents contacts. For simplicity, we have, I guess, only consider contacts of a certain length and certain duration. Otherwise it gets really, really messy. But even so, basically everyone's connected to another directly or indirectly. Usually one step connects them. The most indirect would only be 2 steps, which is amazing. What does that mean? That means that for certain diseases, it's going to spread through the population very quickly. And for example, these 4 individuals in the center, we would consider them to be potential super spreaders. So if any of those 4 get sick within 48 hours, over half of the population will be infected. In 72 hours, all of the population will be infected. A lot of this analysis was done by Megan Kraft. So we collected the data. We were inundated with over 300,000 contact records. And that was more than I had ever dreamed would ever happen. And it took a while, but Megan is someone who's an expert epidemiologist with social networking. And so she handles large data sets. And so the 2 of us worked together to create this analysis. And so this has been published in a series of papers over the last decade. But more importantly is the technology. And so the technology, because we're in the early stages of that, we were able to publish some early papers describing the technique, by the way. So this is actually modeling, if you infect an individual, within 12 hours, you'll have 6 individuals infected, depending again on the pathogen. And then again, within 72, all of them are infected. Here's just a brief list of projects that are now using proximity detectors with the species and then the location. So you can see that they're being used worldwide, and they're being used on a variety of different species. And this is a short list. I didn't try to do an extensive list, but these are just recent studies, and that's had a large impact. So that all started actually in Bussey Woods in Cook County. So that's proximity detectors. So that was in the early 2000s. Since then, beginning in the late 2000s into the 2000 teens, we began using GPS collars. They finally were able to revise them and modify them to the point where they could fit on coyotes. So we were the first study to begin putting them on coyotes, and then other studies quickly followed. We're definitely still one of the few studies using them in an urban setting. So this is a GPS collar, which means that the satellites are locating the animal. What's nice about these transmitters is super expensive, so we don't put them on all of the animals that we capture, but what makes them so valuable is that we can change the rate at which they are being located by the satellites, so we can increase the number of locations when these animals are moving. We can also, the accuracy is much greater from the satellites than when we radio track these animals on the ground. And then of course, we can track them in areas with satellites that we can't physically get to. And that'll be important a little bit later when I bring up another study. So these are GPS collared coyotes within the city limits of Chicago that we've been tracking. I'm not gonna spend a lot of time on the coyote project because I think people have already heard quite a bit about it, but just to illustrate, each one of these colors represents a different individual coyote. Those dots represent GPS locations, and in many cases, especially when you get down toward the center of the city or the loop, but even in some of the immediate outlying areas, the reception and our physical ability to actually get close enough to these animals to track them becomes almost impossible. So we would not be able to do this. In fact, we weren't able to do this in the study. We had to rely on mostly animals out in the suburban areas. We couldn't do this until the GPS collars were actually available and refined enough to be able to place on the coyote. So this has opened up whole new windows into how coyotes are using the most extreme urban landscapes in North America. This is just a view of 4 of those animals that we were looking at earlier. Again, just to illustrate how urban it is. This is on Lakeshore Drive, and this would be the canal over here. And you can see that, again, we get high resolution locations for these animals. And just another image of one of them that we would not be able to track. There's almost no access points along here, and the interference, the radio interference is unbelievable, and yet the satellites are able to penetrate that quite well. That's coyotes, and so we're still continuing to do that work because we're doing different things with them now. We'll be actually evaluating some hazing techniques as well as some other management solutions for these guys. We also were the first to actually apply GPS collars to raccoons. They continued the refinement of the GPS technology, and once they got down to a humane size, we began putting them on raccoons for future use later on that I'll get to in just a moment. So we were the first study to actually use GPS technology for raccoon movements, which are quite a bit different than coyotes. So for example, we focused our early efforts here in Dan Ryan Woods, which is a tiny little forest preserve on the south side of Chicago. It's only about 100 acres, I believe, or less. And each one of these colors represents an individual raccoon, and all of those points represent their satellite locations over a 9-month period. And what's crazy about these guys in this highly urban setting, in a highly urban park, is that they're not traveling anywhere. So again, this is over a 9-month period. This is slightly less than 100 acres, and they're not even using all of the forest preserve. This is a golf course over here. They're not going over to the golf course. These are all animals that are obviously finding food associated with people rather than foraging for their natural food. So for example, here's one of our animals, again, that spent months and months, basically in 3 locations in this, only within the first 2 blocks of the neighborhood. Doesn't even go out any further than 2 blocks. And that's its whole life. And so we see more examples like that. It's going to look quite a bit different here once we move over to Manitoba, let me tell you. So that's GPS technology and getting information from those satellite transmitters. I have to tell you that in addition to just those cool maps, because we can change the rate in which these animals are located, we can actually look at things and measure things such as the velocity that the animal is traveling, the direction, the turning angles. That may not sound very important, but we use those things to be able to characterize their behavior in certain different types of habitats. And I'll give you an illustration of that when we get over to Manitoba. And so that's important for conservation because we can evaluate different habitats based on what their activity is in that habitat. So in 2012, I met a person by the name of Dr. Seth Newsom over at the University of New Mexico. He's a worldwide expert in stable isotopes, and he worked with the marine mammals at that time. And we talked about the potential use of stable isotopes to characterize the diet of coyotes in particular. This is because the traditional method for measuring diet in coyotes was to collect their scat on the roads, because coyotes quite conveniently deposit scats on the road, and forever, for decades and decades. That's how biologists analyzed their diet, was by picking through their scat. But we always felt that certain diet items may not show up in scat very well, and we also don't necessarily know which individual coyote that we're actually sampling. So stable isotopes offer us the possibility of learning more about their diet and more about the individuals. So what we do is we snip a little whisker from the coyotes, and we analyze that whisker for its diet. So in this case, we know exactly which animal we're sampling. We know their age, we know their sex, we know where they live, we know if they're an alpha animal, they're subordinate. We know all of these details that no one had been able to use before in diet studies. So it opened up whole new windows into this really important aspect of their ecology and management. So for example, I've just artificially drawn this really crude whisker, really long blue one here, and just to illustrate, so we snip it off at the base, and then one of Seth's technicians who are really good at this, they section that whisker into identical sections, identical by weight, and each one of those sections is analyzed for diet. So in this case, we're able to actually track the animal's diet over the time period that that whisker has grown. For in the case of coyotes, you're talking about, on average, about 6 months. So 6 months of regular, basically, estimates of that animal's diet. So what are isotopes? So elements that are associated with biological organisms often have isotopes that vary in their structure, and they vary in certain patterns. So those are the variations are called isotopes. And so here are some of the elements that have variable isotopes associated with them. And in our case, we're most interested in carbon and nitrogen, and there are reasons for that. So what's illustrated over here on this graph is carbon is on the x-axis and nitrogen is on the y-axis. Now, I'm not going to get into a lot of detail here, but nitrogen basically gives you a profile of where that animal is located in a trophic level. So in other words, are they a consumer or like a first order consumer? Are they a predator? Are they a grazer? That type of thing. They will plot out differently along that x-axis. Carbon in our world, especially with the animals that I study, that's the really important axis, because carbon is influenced by plant material. And in particular, human-related foods are what we call C4 or corn-based plants. Natural vegetation is C3. And these map out quite a bit differently. And so then when an animal that's an herbivore is eating, let's say their diet is primarily C3 plants, they end up over on the left-hand side of this x-axis. If they're eating, let's say they're eating grain or corn, I mean, people put out corn for deer, all of a sudden that deer is going to end up over here on the right-hand side in their tissue, and it's carried in their tissue. And in fact, there's different types of tissue. All of our tissue retains those stable isotopes for variable lengths of time. So teeth, if you analyze your teeth for stable isotopes, you're actually getting a reflection of the lifetime diet of that individual. If you look at bone, that's years, and you can section different parts of the bone to try and get an idea of whether they're recent years or earlier years, similar to what we do with whiskers. So whiskers you can section, and that represents months of diet. Skin and muscle is more recent, so those only give you information for months or weeks. Blood and some feces for some species are days or hours. Serum, hours, and then blow. I didn't know what else to call this. So this is actually a term for the marine mammals, where this mainly comes from. That would be the actually exhaling carbon dioxide. So when you exhale carbon dioxide, if you capture that in a vessel, you can actually analyze your breath for stable isotopes, and that gives you an idea of their diet within minutes. So we, again, focus on the whiskers because we're interested in more long-term information. Just to illustrate what this looks like, this is actually a recent publication in a big journal, Scientific Reports. This is actually prehistoric information. So you can actually go back to museum specimens or archaeological artifacts, and what's usually used here is bone. And you can look at different species back 15,000, 20,000, 35,000, 40,000 years ago, and look at their diet to see like where did they fall out on this spectrum. So over here in this yellow would be reindeer, prehistoric reindeer. This would be prehistoric bears over in the far lower left-hand side. These are all herbivores. That's the mammoth. And then prehistoric horses, prehistoric cows, or bovids that are related to cows. The predators are over here. And up here, the whole point of this was to look at the last remaining Neanderthals, their diet in the very early stages of modern man. So stable isotopes, again, are super interesting. And what you can do using certain analyses is you can take a deeper dive into these individuals, so these humans or humanoids, and you can actually estimate how much of each of those prey items were reflected in their diet. And so in this case, if we just look at one panel, this is a certain group of Neanderthals. The red diet items, the red prey items, are the ones that were the most important for their diet. So they're making up the largest percentage of their diet. So again, that would be the reindeer and the mammoths. So anyway, that's something that we can also do with coyotes if the diet items are spread out enough. So that was the key here, is that these animals on the isotope are spread out enough to where you can actually do this. So this is what we did with coyotes. We published this back in 2015. At that time, that was the first publication that anyone had published on coyotes with using stable isotopes with their diets. And so again, thinking back to that image that I just showed you with the Neanderthals, here the ellipses are the prey items or the diet items. And then these individual points are individual coyotes. And then where they fall out on this landscape depends on what their predominant diet was. And so what we were able to do in this early work was we could actually create this dotted line using certain statistical analysis that separates animals that are using a lot of human-related foods in their diet. So again, this is that carbon axis. So the animals that are on the right-hand side are using a lot of human food, and the ones that are on the left are using natural foods. And so what we were able to show is that there was variability with these animals. And again, they're different colors because they come from different locations. We could also look at their sex, their age, their reproductive status, all those different things. This is the most recent graph. We haven't published this yet. So this is basically all of the coyotes thrown on that graph. And then this is the last one you have to see for a while. But the important point here, so these are all of the coyotes that we have in Chicagoland that we've been able to analyze. This yellow region represents animals that have human foods that make up over 75% of their diet. So they're relying heavily on human foods. The ones in between the dotted line and the yellow are using human foods over 50% of the time. And then everyone else is just highly variable. Some not touching human food at all, and then others using it to varying degrees. This is the really most complete profile of what it looks like in the Chicagoland area, or for any urban setting anywhere, for these coyotes and their diets. And the take-home message is that they're all there's a tremendous amount of variation going on with these individuals, both across individuals, and then I don't want to get into the statistics too much, but again, remember we're analyzing multiple segments for each individual, which gives us an average dietary point for that animal. And so you'll see a whisker, if you can see my pointer, that emanates out from that point. That's just the variability of the diet along the carbon axis. So for that individual, they're showing a lot of variability. There are others that are not showing hardly any variability at all, which means that they're really keying in on certain diet items for the length of that whisker, which is, you know, up to 6 months. For coyotes, that's pretty amazing. So that's using stable isotopes. That's again, it's opening up whole new worlds. And so we'll look at a couple of them here. So a current study that we're working on is over in Manitoba in collaboration with Delta Waterfowl to try and learn more about waterfowl ecology and especially management to enhance reproduction on the prairie pothole region. So this is an important area for waterfowl reproduction. It's the most important area in North America. It's threatened primarily, well, waterfowl are threatened primarily through two processes. One is habitat loss, so agriculture is consuming more of these prairie potholes in the wetland areas. And then nest predation, especially by mammalian predators and to a lesser degree, avian predators. And so a lot of the research is focused on the predator effect and how much is it impacting waterfowl and what are the most successful ways to mitigate that. And so one of the things that Delta's been doing is drones. And this is something that McGraw has contributed to. And not just the drones, but equally important are the thermal cameras that are fitted on the drones. And thermal cameras allow the biologists to do a better job of locating waterfowl nests for monitoring, and then also locating predators on the landscape. So if this will work, what I'm going to do is shift from PowerPoint, and I'm going to try to shift over to their video, which illustrates the drone thermal cameras. Did that shift for everyone? Are we looking at the YouTube picture?
No luck.
No luck? What if I...
Hi, this is Frank Rohr. I'm president and...
Not playing, you can't see it?
We can
No
only
luck.
hear it.
Shoot, okay. Well, I won't be able to illustrate that. So I tried, Frank. So let me go back to the PowerPoint. So you can see the drone here, and basically the thermal cameras pick up images, heat signatures on the landscape. And the Manitoba landscape is an excellent system to use this technology because it's relatively cold. So it's going to pick up heat. And so it's actually highly sensitive to detecting nesting waterfowl. So they can, again, do a better job of finding nests that they would have missed. And also, so there's always been a question about nest monitoring and to what degree do the biologists bias the results by perhaps finding a nest more successfully in certain areas than in other areas. The drones allow them to be less biased in their monitoring. And then it also locates predators more effectively, obviously, than the biologists. Let's see. So this is a picture of Frank Gehrt. He's the lead scientist and lead, I guess, the president of Delta Waterfowl. He's not CEO. Here's a couple of pictures of the thermal images, the heat signatures that they can pick up. So that's an exciting development in terms of waterfowl ecology. One of the things that we're doing is using the GPS technology that I described for you earlier for raccoons in this very different kind of landscape. It turns out that based on the nest monitoring that they have done, Delta is determined that raccoons are probably the most important, at least mammalian predator of these waterfowl nests. And so once Frank saw the maps we're able to generate of the really high resolution movement patterns of raccoons, we determined that it would be beneficial to not only learn more about how raccoons are exploiting that landscape, but also perhaps to use that information to better trap and remove raccoons. So here are some of the raccoons in Manitoba getting fitted with those GPS collars. That's Charlotte and the graduate student working on the project for Delta. And here's that agricultural prairie pothole landscape with the GPS locations of the raccoons. And we do have the scale presented down there, but it's hard for you to tell. This is a massively much larger scale movement by these animals than what you were seeing earlier for those raccoons. So these guys are going over hundreds of acres, actually in some cases over 1,000 acres in their movements, as opposed to, again, those urban raccoons that were barely going 2 blocks. These guys are covering huge grounds. And so these yellow dots represent the GPS locations of a single raccoon during the nesting season. And you can see how that raccoon is localizing its movements along the edges of the wetland areas, in addition to some other sites that we have to ground truth to determine what are the other attractants that are keeping that animal on that landscape. If we can identify those things, that could be a useful management tool to either remove the animal or remove the attractants, which is a more long-term solution. And then I mentioned earlier that because the GPS technology allows us to look at these movements in such great detail, we can actually characterize their use of the landscape based on their behavior. So this is actually Charlotte's analysis, her research, and this is just one raccoon on a particular portion of its monitoring, and different colors represent different behaviors, again, using different parts of the landscape. So it's probably hard to see the blue locations. That's when the animal's actually traveling quite a bit, which is something we rarely see in the Chicagoland area. And that's where it's using different areas than it is while it's foraging. And then I mentioned stable isotopes and how we're using those for coyotes. We're also going to use this for this system, this Manitoba system. Here are all of the potential predators of a waterfowl nest in Manitoba. They've all been documented having some effect, some level of predation on waterfowl nests, and including crows, ravens, and magpies. And so we are collecting the hair samples for these individuals so that we can actually see which of these predators are most important in terms of shifting their diet over to waterfowl nests. No one has actually done this before, and definitely not with stable isotopes. No one's ever looked at the community of predators in that system all at once to see who's shifting and who isn't. That's important both ecologically for us to understand that, but also from a management perspective, it helps us to refine our management techniques to focus on those that are most important. So even though raccoons were the ones that were often seen at the nests, there could be important predation that's being missed for one reason or another. And by us actually collecting the hair of these individuals, we can track their diet during that nesting season. And so again, perhaps you provide better data to focus management activities. This is a study that we did a few years ago and that you may or may not be aware of, but it also used a combination of GPS technology and stable isotopes that actually had important management implications. It had implications in a lot of different ways. So this is Cape Breton Highlands National Park over in Nova Scotia. It's off the coast of Nova Scotia. Parks Canada had an issue that occurred in that park, and they actually asked McGraw to use our expertise to evaluate the reasons why the incidents were occurring and to develop a management plan. The incidents were attacks on people by coyotes, and there were an increasing number of violent attacks by coyotes on people in the park, despite the park attempting to remove coyotes. It was still continuing, and unfortunately, the most extreme was the only adult fatality that's associated with the coyote attacks was Taylor Mitchell, who was killed in 2009. She was attacked on one of the most popular trail actually in the park. She was hiking alone, and otherwise it was an unprovoked attack. And in fact, there had been no reports of coyotes acting bold or aggressive prior to that attack. These were the coyotes that another couple on the trail took pictures of them about 15 minutes before they attacked her. So first we used GPS technology to determine what was the ecological system there. If coyotes will scale the area of their movement based on the resources. And in fact, if they're finding human food and becoming reliant on human food, they restrict their activities quite a bit, kind of like what you saw with the raccoons. If they're not, if resources are limited, their home ranges are going to be quite large. And so this is a map of Cape Breton Highlands, and here are some of the animals that we radio collared. And this is where GPS technology was crucial, because we could not use standard radio telemetry in this area. There's only one road that goes along the perimeter of the park. There was absolutely no way we could physically radio track these animals. It had to be done by satellite. And it actually was a really important finding because their territories were huge, as large as 145 square kilometers. It's massive compared to Chicago. So for comparison, here are some GPS collared coyotes that were located in Poplar Creek. That's the same number of packs as what we had over in Cape Breton. But the scale of the map is completely different between the two. So if I change the Chicago one to the same scale as Cape Breton, this is what it looks like. So this is how large the territories are of the coyotes in the Chicagoland area versus Cape Breton. Night and day difference, which is an illustration of limited resources. And in fact, that was consistent with other things that we saw, such as really light body weight and low reproduction. When we use stable isotopes, again, we were really interested in the anthropogenic use, because if coyotes are attacking people, often, in fact, the park was getting criticized by people that aren't there, especially pro-coyote people, because they were saying that the park wasn't doing a good job of managing human food. They thought they were, and in fact, when we were there, they absolutely were, and it's reflected in the diet. So here's that anthropogenic line, that dotted line. These circles are the coyotes that we recorded. There were only 2 coyotes that actually drifted over to that anthropogenic line. All the rest were clear down here with moose. And in fact, that was what we were seeing. The meat was moose while we were there. And I don't really have time to describe this too much, but these are actually segment lengths of those whiskers of 3 coyotes over here from those Cape Breton animals, and 3 coyotes over here from those Cape Breton animals. We're only looking at the carbon axis over here on the left. So we've basically flipped the graph. And so now the anthropogenic line is horizontal with a 95% degree error rate. But when you see each one of these animals drift into that area, that means for that segment, for that period, they were eating human food, and then they would drift out. So you can see that none of them were using human food regularly. These were the only 3 of all of the animals that we trapped that actually drifted over anywhere close to human food. These are completely down by moose, and you can see that in contrast to the tremendous variation in diet reflected by coyotes over here, these guys weren't their diet at all. And in fact, it was all moose throughout the whole length of their whiskers, so for months and months. But these 3 were the ones that attacked Taylor Mitchell, the ones that were not eating human food. And this is the only case, obviously, where anyone has ever measured or published any kind of diet information on coyotes that have seriously attacked someone, or in this case, killed her. So this was totally surprising, but again, once you understand the whole system, it's just a very extreme system, and these animals are actually attacking moose as part of their diet. They also are not hunted or trapped at all in the park, so they have no negative experience with people. So the combination of no negative experience with people and having to pursue large prey then probably led to them attacking a person. So, by the way, I should mention this was important, and this had important management implications, because most of the national parks in Canada are very reluctant to lethally remove animals from their parks, only if in extreme circumstances. But because of these results, one, we were able to demonstrate that they were already doing an excellent job at managing human food in the park. They're just, they do a really good job with that. But the second thing was that they needed to be more aggressive and less tolerant of bold behavior by coyotes. So they changed their tolerance level, their management strategy. And since we did this work, they haven't had any attacks by people, but they do now remove coyotes. They also are actively talking about maybe perhaps opening up the park for some limited types of hunting. They're already doing it to a limited degree for moose. They may do it for coyotes as well. So real quick, in the time that we have left, so white-tailed deer, I need to get to the vaginal implants. So white-tailed deer obviously are a management challenge in urban areas. Many areas, especially in urban settings, or even natural certain parks, need to control deer either through hunting or by sharpshooting programs. But often when they propose a hunting program, the agency or the parks, they need to be able to justify it based on some target goal. Many deer need to be removed. Well, the models that they use to project the deer population estimates as well as their response to removal use fawn survival. It's one of the critical components to that that model, but often they don't know what fawn survival really is. We definitely don't know what it's like in things like Cook County forest preserves or other large metropolitan areas. So since 2013, we've been collaborating with the Forest Preserve District of Cook County to estimate fawn survival and identify important causes of mortality. We definitely are interested in to what extent are coyotes removing fawns, and does that cause an adjustment in their population models. So this is very difficult to do. There's a lot of moving parts to this. We have to capture does, even though that is us, and that's actually Shane there, but that's a buck. So we can't always pick and choose what we catch, but when we catch a doe, we determine if she's pregnant, and if she's pregnant, then she's radio collared, ear tagged, and then we insert a vaginal implant into her uterus. So this is that vaginal implant. The wings here are flexible, so you have to use an implement to get it up past the cervix, which is pretty uncomfortable for everyone involved. But you can get it up into the uterus, and so then it will stay there with the fetus until the doe gives birth. When she gives birth, the transmitter comes out with the neonate, and when the temperature changes, it activates the transmitter so that we can determine when a birth has occurred. And then we can get out there, hopefully before a predator does, and radio collar the fawn. So here's a fawn that we located, and we do this very quickly, so we can basically get the fawn. Obviously, we're very careful about scent, so everyone wears long sleeve shirts, gloves, that type of thing, and we weigh the fawn, age it, and radio collar it, and always in less than 5 minutes, usually with much shorter than that, just a few minutes, and then we get out of there. That's an animal that's about to get the collar. So these collars are specially made so that the stitching is loose, and so as the deer grows, the stitching pops, and so the collar actually grows along with the deer, so it doesn't hurt the animal at all. Here's a picture of one of our does with the fawn radio collared, and there's a lot more to this than what I have time to go through, but this is extremely labor intensive, very difficult to do, as I mentioned earlier. Through 2018, we collared 81 fawns. We had 55 mortalities. These are survival estimates of the fawns each year. And the main thing I want you to notice, so this is, for example, in 2013, that would be roughly about 20% annual survival of the fawns. And the next year would have been a little over 60% annual survival, and then it dropped back down. And then you can see all the way down to 2018, it drops down lower than 10%. So in general, survival of fawns is fairly low. And again, this is a Cook County Forest Preserve. The primary cause is coyote predation. Coyotes are the only predators we've documented taking the fawns. And this is the number of weeks post-birth. So in other words, the age of the fawn, if you want to think of it that way. And predation takes place primarily in the first 4 weeks. Again, it's all coyote related. After that, there's almost no mortality whatsoever. So if they can get past that first 4 weeks of life, they will have a greater than 90% chance of surviving the rest of the year. But it looks like coyotes, at least we've monitored them in 2 forest preserves so far. Coyotes are definitely having an impact on recruitment of deer in these forest preserves. And finally, really quick, with river otters, this is a relatively new project. We're using plants. So this is an animal that was live captured in one of the forest preserves. It's going to get an abdominal implant, and that way we can track them because they can't wear collars, they can't wear anything externally, and then we also use remote cameras. So remote cameras are an important aspect because it's very difficult to capture otters, and we're still in the early stages of this. So here's an otter that had gone through the surgery and is being released. And so we've done that for 5 otters so far. Here's one of them that we were able to get a look at. So there's photographers that follow these guys. The nice thing about otters is that they're active during the day as well as at night. So you can get some good visuals on them. And then these are the remote camera monitoring. So Shane has been doing a fantastic job of distributing cameras across Cook County and attempting to work out their distribution in the Chicago land area. And then we focused our trapping once we identify where these otters are. Here's a picture of 3 otters together in Crabtree Forest Reserve. And here's a coyote watching one of the otters, again in Crabtree. So I think with that, I'm gonna probably end it. Those were critter cam images. So we worked with National Geographic to develop critter cams that actually will follow the eyesight of the coyotes, so coyotes can actually film themselves doing certain things. So I wanted to try and compete with Frank for the video, the drone video, but since that didn't show up, I'm gonna play fair and not do that. And plus I'm at the end of my time. So with that, if anyone has any questions or comments, I'm happy to entertain those.
Stan, would you stop the screen share, please?
Yes. Hold on. Oh no. 1 second.
Well, to start with, Stan, I know this is something that is sort of the end result of our work in Manitoba. What would be a possible outcome that would affect waterfowl production from your work identifying raccoon predation and the level it affects waterfowl success?
So one of the possible outcomes, and we've talked about this, is that some of the early results, I'm sorry, oh, there it is. There. One of the early returns is that it looks like the raccoons and probably other predators as well are closely associated with human structures or human alterations to the landscape. So often these could be abandoned buildings. So it turns out that abandoned buildings are actually pretty common on that landscape. Old farmsteads or barns, that type of thing, chicken coops, even things such as culverts can provide a refuge for these animals. Again, it's a very desolate, kind of open type of habitat, and so any kind of cover that you provide them, they take advantage of that. So one of the possibilities, and that's been reflected in some of the GPS data, so we've suggested that perhaps some long-term solutions may be used in addition to predator removal, would be to actually remove some of the leftover human artifacts, I guess, on that landscape and kind of clean it out a little bit and take away some of the den sites, especially during the spring. For example, raccoons, foxes, coyotes, skunks are all raising young during the same time that waterfowl are nesting. If you remove those den structures or cover for them to raise young, then that has more of a long-term solution perhaps than predator removal.
And more efficient.
More efficient, right. You don't have to do it every year. So whereas with predator removal, what they're finding, what Delta has found, is that you have to repeat the process nonstop. And if you don't do it, you start back at square one.
Thanks. I think one of the things that is not apparent by Stan's speech, but when you talk to him, is that even though technology has advanced us tremendously. This work is on the backs of some incredibly dedicated people, because I learned this the other day from lunch with Stan. Just strapping a collar on a raccoon is not so easy. And it can take weeks to get enough raccoons caught and collared for us to have a decent level of confidence in the results we get. So, we look at all these high-tech things, but it still comes down to people like Stan and the students and scientists he work with, are also fostered by McGraw and members like yourselves to do the work on the ground, which I don't think that'll ever entirely go away, even as we're advancing by leaps and bounds with what we can do with technology. Stan?
Yeah, absolutely. Some of you, you probably haven't had the pleasure of placing a collar on a raccoon, but so you probably don't know what's so tough about that. Well, obviously they're immobilized, but the biggest thing is that raccoons have no neck. They have no neck at all, especially at certain times of the year. And so it actually is kind of a challenge to again, correctly place a collar on the animal so that it actually stays on, and then it's also humane for the animal. So actually a lot of skill involved. So most of my PhD was spent learning how to radio collar a raccoon, probably.
Maybe when you have your next in-person speech at McGraw, we can have a raccoon collaring contest.
That would be interesting.
Yeah, good sport.
So, yeah, the same problem occurs for skunks as well. So
I think we'll pass on the skunk collaring contest.
Skunks have no necks whatsoever, and they're much smaller to work with. I didn't touch on that. We actually did. This is before I left for Ohio State. We did a 6-year study at McGraw on skunks. It was by the request of the IDNR.
Yeah.
They were actually concerned about the skunk population, again, for disease reasons. So I agreed to do it because we want to be a good partner with the DNR. But I thought, there's no way I'm gonna, you talk about the technicians, the students, I thought I'm gonna lose all of my technicians and my students when we start doing this skunk work. And especially it turned out that skunks actually become very trap shy, and some of them refuse to go into a trap at all. And our goal was to radio collar all skunks in an area. And so we would go out at night radio tracking them, and I would see these skunks out there, and they weren't collared, and yet we couldn't catch them in traps. And so I finally got frustrated one night. I saw one, I said, dang it, I'm just gonna catch it. So we just jumped out of the truck, went out and caught it. And that turned out that was the most effective way to actually trap or capture these animals. And I thought, well, there's no way these techs are gonna go for that, because you're gonna get sprayed. And it turns out the bottom line is that of all of the projects that we've done over the years, the only one that I've had to do conflict resolution on among the students and the technicians is the skunk project. And it's not because they didn't want to do it, it's because they did want to do it. And if they were getting ripped off on their opportunities to go out skunking, they would start complaining about it. So they actually fought to be able to go out at night and chase down skunks. So, because it was so much fun.
Perhaps we're missing out on an outdoor recreation opportunity at McGraw.
Yeah. By the way, in terms of technology, one of the things that I did that still hasn't been done by anyone was that we were trying to figure out why was the skunk population not rebounding the way it should. I know that most people would say good, but the DNR was concerned about why is it not rebounding, and I was concerned that the neonates, the young animals, while they're still with the mother, we were missing some important information there. I thought that there was some mortality occurring. So no one had ever tried to put a transmitter on baby skunks. And so I actually had leftover transmitters from a bat study that we did. So we used surgical glue glue, and we could weave it, we could stitch it into their fur and glue it so it's temporary. And so we were able to track the little babies as they're traveling around with their mother and actually follow their survival, at least for a couple of weeks. And then it would fall off, then we would replace it again. So we actually were able to get an insight into a part of their life history that no one had done before. And by the way, baby skunks, cutest things ever. Coyote pups are pretty cute. Raccoon cubs are pretty cute. Baby skunks, cuter than all get out, and they don't spray. They just sit there nice and they just look at you and you want to take them home with you.
That's awesome.
Stan, thanks a lot for this presentation. One of the questions you didn't cover, bobcats. And we recently found a bobcat on a trail cam that had a collar on it. We asked around, and it turns out that we're about 40 miles south of the border with Wisconsin, and they've done a collaring opportunity, and it apparently traveled south and probably took off even further south. But it's kind of interesting. I don't know if you're going to be looking at bobcats or bobcats interest you. I don't think it's a major major problem for predators, is it?
No, they really aren't, for the most part. They'll take turkeys to a certain degree. So that's sometimes the turkey hunters will complain about the bobcats. That's actually one of the easiest ways to trap them is to use some turkey feathers. But no, we would love to put some collars on some bobcats. And every once in a while, we'll have a bobcat show up on a trail camera somewhere in the forest preserves. But before you can actually start trapping that area, it disappears. So there's a lot of questions about, can bobcats adjust and adapt to an urban landscape like the canids, like the coyotes and the foxes and the other things. And so there's been some really nice studies done on bobcats in LA and in Dallas, where they were able to show that, bobcats are having litters on top of people's houses.
Wow.
And underneath decks, but also on top. But LA is different. So both of those studies were able to show that to a certain degree, bobcats could adjust, but those are the only ones in the US. There's basically little to nothing known about how those cats adjust or don't adjust. They have a limitation. They have to eat meat. They don't have a choice. Most of the other species that do pretty well in an urban setting, at least they can shift over from meat to human foods if they need to, whereas that's a struggle for cats.
So, incidentally, we did have a bobcat turn up on the McGraw trail cams this past fall, but only seen once and not since. So they're highly.
Oh, sorry. Hi, I had a question too, Stan. I wanted to ask about, I came in a little bit late, so I don't know if you talked about this, but microchips I think that, microchips are used to some degree, but maybe just to identify, like you do a lost dog. But is there any way to make, like, instead of having to put a collar on an animal, have a microchip be able to, receive satellite information or something like that?
Right. So you're right. So we use microchips for the pups.
Okay.
We also microchip some of the adults as well, but the pups get microchips because they're obviously too small for radio collars. And so that way they're permanently marked, and each one's individually marked, just like your dogs are and your cats. And that way, when we catch them later, then we know who they are. We know who they were born with. And then we can put a collar on them. The technology definitely exists to use satellites to track the microchips. So it's there, and it's used for some species. It's not successful yet for the ones that we study so far. We're waiting. Some of it, the early stuff, the really small satellite tracking, again, as I said, the technology exists. We know that the military is able, and Homeland Security, they're able to do that. But there's often a delay. Well, there's always a delay from getting their technology down to our use. Same thing was true for GPS collars years ago. Same thing was true with GIS mapping. Now the military had to unrestrict it. So hopefully we'll be able to do that before I retire, but we'll see.
You're not retiring, Stan.
Right, because I'll probably pass away
No.
before I get there.
Any other questions for Stan?
I've got one. First of all, thank you for the information on the bobcats and turkeys. It gives me another excuse as to why I sat outside all day today and came home empty-handed. So thank you. I've got an excuse now. Yeah, no, I appreciate, that most of your work has gone into data gathering and using technology in that way. But have you started to use or to try to develop techniques in using technology to manage wildlife? I mean, given the number of urban interactions and what's reported mostly as negative or aggressive behavior of coyotes now, have you been using technology at all to try to come up with ways to manage that? Come up with hazing methodologies using technology or anything like that?
Yeah, so we've tried to evaluate hazing for a number of years, and using more traditional radio telemetry, and it just never really worked very well. Now with GPS collars, we're going to attack. We're going to attack it again, starting in the next month or two. So we're in the process of designing those at the moment. So the tricky thing though with hazing is that it's always going to be met with mixed success with coyotes. Like every other management technique for coyotes, it's going to work well for some individuals, and then for others it won't work very well. But we can still do a better job of documenting, the circumstances when it does work well. So we're going to do that. We have used our technology for management. So for example, the early years of the coyote work, almost I think by the second or third year of the study, the animal control agencies, not just in Chicago, but around the country, started adjusting their management plans from where initially, if a coyote was seen in an area, they would initiate trapping to remove it, again, in an urban setting, because the belief was that coyotes were either not supposed to be there, or it was a transient, or maybe it was sick, because it wasn't natural. They just shouldn't be there. But our research showed that two things. One, they adjusted, and so now it is kind of it's a definitely normal thing for them to be there. But secondly, when you do the removal, we've been able to show through technology exactly how long that removal is going to last before they're replaced by another animal. And so we were able to actually show with data to the management agencies, this is what happens when you put all that work and effort to remove an animal that actually was just seen crossing a road, perhaps, or especially railroad tracks. You can make the decision. We don't tell them what they should or shouldn't do, but here's what's going to happen. So you put that time and effort to remove them, and that's going to last about a week or two. And then you're going to have another coyote that'll replace it. So once they saw that, they adjusted their management plans to then focus more on animals that were actually exhibiting behaviors that would actually justify their time and effort and the money to get them out of there, to remove them. So it saved them a tremendous amount of time, because prior to that, if they got a call from someone that was uncomfortable with seeing a coyote in a park or in a cemetery or something, they were obligated to at least try to remove it because they had no other response. Our data gave them the ability to then educate themselves and then also educate the public that, it's not always necessary to remove an animal if you happen to see it there. So that's an example early on. There's been some others where our data have been used for actual management. And of course, I gave you the example of Parks Canada. I mean, that was a huge deal for them. And so far, they haven't had any attacks on people. And that was what they needed to see, was that being too tolerant of these animals was causing a problem, is putting people at risk. And the level of risk is much higher in that park because of that system than it is, say, here in the Chicago region, where resources are plentiful and coyotes aren't attacking large things.
Well, absent any other questions, I held off on introducing McGraw's new Senior Vice President, Crystal Shaw, who is with us tonight. If you've not been out to McGraw to say hello, please stop in. We're open for business. And Crystal, we're so happy that you're with us. I was waiting, hoping more people would join us. But
Thank you.
we're so appreciative to Stan, of course, but most of all, we're appreciative of you, our members, who support Stan's work. And the other unspoken thing is, you saw Dr. Milling, you saw some of the other scientists like Shane, who Stan has mentored, guided, sponsored their own research. Through Stan's work and because of your support, McGraw has built an unparalleled network of wildlife biologists across the country who are carving out their own stories thanks to Stan's mentorship and your support. So we are talking about what McGraw is currently doing, but when you think about the family tree, McGraw's reach has spread across the country, across the continent, and it only continues to grow because now some of the students that Stan taught have their own students. And it's all because of your support and your ability to help us do what we do. So we can't thank you enough for that. And we can't thank you, Stan, enough for sharing your time and your expertise. It's always a pleasure.
It's my pleasure too.
Thank you, Stan.
Thank you, Stan. Thank you very much.
Thanks to you guys.
Everybody,
Thanks, Stan.
come out to McGraw. We're open.
Yep.
Bye now.
Thanks,
See you guys
Gary.
later.